Okay, great. So I hope you can see me and also hear me well and see my slides. So let's begin. Yes. So the title of my talk is Infusing Structure and Knowledge into Biomedical AI. The code base and tutorials, data sets of projects that I will be highlighting throughout the talk are available from our lab website. So if you go to zitniklab.hms.harvard.edu, you will find lots of resources that support the projects that I will highlight throughout the talk. So I hope that that can also be useful educational material for those who are interested. Okay, so let's begin. Science in biology in particular and medicine have over the past several centuries crucially depend on existence of scientific instruments. And so in the past, those scientific instruments that allowed scientists to measure and observe a natural phenomena have primarily dependent on physical devices. Those were physical instruments that have facilitated discoveries. And nowadays, we are in the era of modern data-intensive scientific disciplines, and we need new kinds of instruments that I believe will be in vital ways depend on knowledge discovery from large, big interconnected data sets. So with that in mind, an important question to ask is what makes then biomedical data so unique or challenging? What are distinguishing features of biomedical data that make it different than data that we encounter in other areas of scientific research? So I would argue that there are several important considerations to have in mind when developing and applying algorithms and methods to biomedical data, including the following. For most problems of interest, there is not necessarily a single data set that can fully capture the entire complexity of phenomenon. Because of that, there is typically a need to integrate heterogeneous, confounded data that sometimes span multiple levels of biological organization, going from molecules all the way to society. And we really need to integrate then data at these multiple scales to understand the particular phenomenon well. And so this is in conflict with the more classic view of machine learning in AI that primarily assumes that the input data is a fixed input that nicely fits into a single large flat data matrix. In many cases, we have really multi-scale heterogeneous confounded data that we need to work with. Second point is often there is a need to translate statistically accurate predictions into actionable hypotheses. Actionable hypotheses for me are those that can lend themselves immediately to downstream experiments or that can directly inform clinical research. And so that means that we need to think about other important properties that our algorithms have and need to find ways to take black box models, make them more like white box models that can be interpreted meaningfully. And why is that so important? That is important because often computational tools that we're developing are really used by biologists, used by other scientists, used by clinical researchers that are using those tools to make important high-stakes decisions. In clinical settings, that those might even be decisions that regard life or death. In scientific research, there might be decisions about what expensive, costly, and resource-intensive experiments to run. And so because of that, it means that what we need is algorithms that are not only accurate, but that are robust, that behave responsibly, that are trustworthy. We need to have checks and balances built into deployment of these algorithms. And those models often need to provide, in addition to predictions, explanations or some form of interpretations that can inform downstream decision making. Finally, that's a final point for today that I think it's also very important for biomedical data is that, well, we have seen tremendous amount of discoveries and breakthroughs that AI has brought on in a variety of disciplines over the last decade or so. And most of these recent breakthroughs in AI have really depended on lots of labeled data, on models that can be pre-trained on very large, well-annotated and labeled data sets, and then those models achieve broad generalization across a variety of downstream tasks and a variety of downstream settings. I'm mentioning that because this is in sharp contrast with what some of problems at frontier of scientific research and frontier of biological research are characterized by, where they would typically have very little labeled data points that can be used to train models. And so that means that we need novel ways about developing models that go beyond standard supervised learning frameworks. Throughout this talk, I will offer some of solutions or some of our ongoing projects that address these unique challenges of biomedical data. The overall approach that our lab is pursuing is that of realizing that in order to be able to bring heterogeneous data together, to make sure that the models we built are trustworthy, and to make sure that they can be trained and perform well in challenging settings where labeled data is scarce, we really need to find ways that integrate heterogeneous, that integrate knowledge and structure into our machine learning models. And that allows us then to model interactions at the molecular level in the form of interactions between molecules such as proteins, the ways those molecules, when they are therapeutic targets, are targeted by external agents or various kinds of chemical perturbations or small compound molecules, which might then interact further within each other, as well as with the diseases that coexist within a given individual person, and all that is embedded within the broader local environmental community. So networks or graphs allow us to bring this additional knowledge and structure into models and address many of these challenges that biomedical data bring. For the purpose of today's talk, the key algorithmic innovation that I'll be focusing on that has brought on many areas of success and might be relevant to several researchers on the call who have talked about projects regarding therapeutic innovation, modeling of ontologies, questions related to single-cell biology, multi-omics data integration. These many areas of success in the recent years has been fueled by advances in a particular area of AI that is known as graph representation learning. So graph representation learning is a class of algorithms. It's a field that provides tools that allow us to learn, reason on large interconnected heterogeneous graphs or networks and derive predictions, patterns, insights from that data. In a single slide, let me try to kind of explain the gist of what of graph representation learning as a field. So this is really high level summary, but the most important kind of concepts here are that related to graph neural networks. So graph neural networks are a class of neural network architectures that consist of a series of nonlinear deep transformations and can take as input any graph structure, the data set, such as the one shown in the left, and learn powerful representations, embeddings shown on the right that can be then used for the various downstream tasks. So imagine that as our input, we have this very simple toy illustrative graph that I'm showing on the left part of the slide. And that graph is described by a set of vertices or nodes, and lines between those circles or nodes indicate edges, links. So a graph neural network, F, is then a nonlinear, highly parameterized function that will map each and every node in that input graph, say node U and V, and it will map that node into a vector, which we typically refer to as an embedding. That vector is a point in some d-dimensional embedding space. And so the way graph neural networks do that is in the following way. So that two nodes U and V in the input graph who have similar local interaction neighborhoods or are proxy or are found nearby in the input graph, they will be mapped to points that are nearby in the embedding space. So that essentially means that function F, this graph neural network, is optimized such a way that it generates an embedding space and that performing linear algebra operations, meaning dot products on these embedding vectors, will reflect the structure of the input graph. So there are a variety, There are a variety of different architectures that are in the field of graph neural networks, and a broader implications of these advances is that with graph neural networks now we can apply deep learning much more broadly than what was possible before, not only to medical images, where we have seen architectures such as convolutional neural networks being very successful, and biological sequences where we have seen architectures such as transformers, LSTMs to be very successful. We can now apply graph neural networks to any interconnected graph structure dataset. So this creates some very exciting new opportunities in biology and medicine that I would like to spend the rest of today's talk focusing on. So my plan for today's talk is to give 3 vignettes. And those 3 vignettes are shown here in this slide. We'll first talk about how graph neural networks and graph representation learning more broadly can help us realize rapid therapeutic innovation and advanced therapeutic science. We will then proceed with another vignette into modeling and understanding of interactions between drugs and chemical compounds, and understanding unwanted side effects in patients. And finally, I will conclude with a point on the need for robust AI infrastructure and modern data management resources that can support bioinformaticians and computational biologists and AI scientists to work together on really important problem. And so that will also include many resources that can help the students on the call try out some of the models that I will present in the first 2 parts of the talk. So let's start with the first vignette on rapid therapeutic innovation. So what do I mean by rapid therapeutic innovation? So traditionally, the process of designing and developing new drugs from scratch consists of a very complicated and lengthy resource-intensive process. It takes on average between 12 to 16 years to develop a new drug from scratch and deliver it to the market. And it can cost $1 to $2 billion on average for each and every new drug to be developed from scratch. So this traditional iterative development, experimental clinical testing, and approval of new drugs is sometimes not feasible, in particularly in cases where there's a need, there's public health emergencies that require rapid development and deployment of safe and effective therapies. There is a need to really respond quickly and be able to provide those safe, life-saving therapeutics to patients. So the challenge is it possible to compress years of work into months or even weeks through the use of AI, automation, and new data sources? The answer to that is yes, and I will illustrate this answer through a project that we have done here in Boston as part of the effort to identify safe and effective therapies against emerging pathogen early on in the pandemic. And so in particular, the approach that I'll highlight and the project the core of it took place between March 23rd of 2020 and June of the same year. And during that span of 3 months, we were able to bring together a team of state-of-the-art sort of scientists building state-of-the-art machine learning algorithms that were implemented and applied to identify therapeutic opportunities for COVID-19. And those predictions made by models were directly fed into experimental effort and screening to identify chemical compounds and drugs that would inhibit the virus. And so that really has been done in such a short period of time, primarily because of the opportunity that new data resources and AI has enabled. So as I mentioned, that the discovery and development of new drugs from scratch is a very expensive and resource-intensive process. An enticing alternative to it is known as drug repurposing or drug repositioning. So the idea here is the following. Instead of designing drugs completely from scratch, would it be possible that we would start from some of drugs that are already on the market and that are used by patients, albeit for different diseases and have different indications? Or could we take drugs that are not yet approved, but that are in late stage of clinical development, and can we repurpose those drugs for novel use? So think of it as taking these old drugs or a bit better understood chemical compounds and find if they can perform these new tricks in such a way that would be useful for novel diseases and emerging pathogens. And we would want to do that because that can help us identify safe and effective therapies, possibly much faster and at much lower cost. So in the context of the study that I'm highlighting here, that was focused on the COVID-19 as an emerging pathogen and disease that was very pressing at that time, the first question that we had was, well, how can we even computationally represent that pathogen? And so the way we approach that is by leveraging the data generated by Niven Krogan's group from UCSF, who expressed 26 out of 29 SARS-CoV-2 proteins and used a mass spectrometry to identify over 300 human proteins that the virus attacks. So those set of 332 human proteins that are bound by viral proteins were used as a representation or construction of a network module that represented COVID-19. We took that module and that set of 332 proteins and interactions between them, and we overlaid it on the human interactome. Human interactome is a graph, it's a network where nodes are proteins and edges encode physical contacts between them. And so COVID-19 disease module was a computational representation of COVID-19 identified as a subgraph induced on the interactome. We also induced on the same interactome a large number of drug modules. For each and every approved drug in the US, we took its primary targets and we defined a modular subgraph for a drug by inducing a subgraph of the human interactome on primary targets of a drug. And we did it for many, many approved drugs. The question then became, are there interesting signatures or relationships between the COVID-19 disease module, shown here in gray, and drug modules shown in green on this schematic that one can leverage in order to prioritize possible drug candidates for COVID-19? So the data set and experimental setup for this project was the following, has the following structure. It consists of the underlying human protein interaction graph on which we overlaid a large number of approved drugs and proteins that each drug targets, and as well as included information on COVID-19 disease module and corresponding disease module of many other diseases. The task was then given information about drug treatments for already approved drugs. Can we leverage that information and transfer it such that we can identify candidate treatments for COVID-19? And so why is this challenging? This is challenging because there were no safe and effective treatments for COVID-19 at that time. The pathogen was very poorly understood early in the pandemic. So from the algorithmic standpoint, the challenge was, well, we had to have an algorithm that would be able to make predictions for a class, for a label, yet there are no examples of positive examples that one could use to train a supervised learning model. And so that is a challenge for prevailing methods that require abundant label information, supervised signal to train those models. And labeled examples were scarce in this setting, and they were scarce in many are also the same problem arises in many areas of scientific discovery. So the solution to this problem is really to use algorithms that allow us to train predictive models in these hard settings, and they're known as few-shot learning algorithms. So let me just quickly illustrate what few-shot learning is concerned with. So the idea is that we have a problem that we want to solve with a machine learning model, but we have very few labeled examples to train a model. Those labeled examples, when they are very scarce, they're called shots. So few-shot learning is essentially saying, can we learn in certain situations where we have only small few labeled examples? Imagine here that we would want to train a classifier that can distinguish between objects of different kinds on images. So the few-shot learning would proceed in the following way. It consists of two steps: meta-training phase and meta-testing phase. In the meta-training phase, we would challenge the machine learning model to learn how to quickly adapt to a new task with very limited labeled example. Perhaps in training task 1, we would challenge the model to be able to detect 3 kinds of objects in an image, having only 2 labeled examples, 2 labeled images for each class. And we would do the same in training task number 2 and training task number 3 and so on. And the model would get better and better, so that at test time, when we would need to build a classifier, duck versus dolphin versus chicken classifier, a few-shot learning algorithm would be able to do that, having only seen 2 labeled examples of ducks, 2 labeled examples of dolphins, and 2 labeled examples of chickens in its lifetime. Well, while we were not interested in classifying images, we were interested in identifying drugs for COVID-19. So we really wanted to use few-shot learning idea on drugs. So the way we approach that is that in the meta-training phase, the algorithm was exposed to different diseases. And each time, each task was defined around different disease, and the algorithm was asked, can you learn how to quickly distinguish between drugs approved for disease 1, drugs not approved for disease 1? And the idea was that when the algorithm solves training task number 1, its parameters are used to initialize training task number 2, and this is repeated many, many times for all diseases for which some treatments are available. And that means that at that test time, that algorithm was able to prioritize drugs for COVID-19 solely based on the knowledge about COVID-19 disease modules with no examples of successful treatments. Predictions made by the algorithm were first evaluated against a set of drugs that were considered in clinical trial, and the results of that analysis is shown on the left part of the slide. And given the promise of those results of performance of the model, we wanted to understand whether predictions would really identify compounds that would inhibit viral growth, as drugs that are considered in clinical trial are really only candidates. They are not drugs that would be proven already to be safe and effective. So predictions made by these AI models were then considered by National Emerging Infectious Disease Laboratories in Boston, who took top-ranked drugs predicted by the algorithm and experimentally screened them on African monkey cells and then later on human cells. So without going too much in detail about the experimental work here, the takeaway message is that over 918 predicted compounds were screened for efficacy against the virus. And by screening these top-ranked drugs, we obtained a near 62% success rate, meaning that 62% of drugs that were predicted by the algorithm and experimentally screened in the wet lab were shown to inhibit viral growth, in contrast to what would be possible to obtain if the screening process in biological lab would not be guided by AI predictions, where the success rate would be around less than 1%. So that's an order of magnitude improvement over non-guided screening when looking at the very top-ranked drugs in predicted list. A natural next question to ask for us was, well, is there something we can learn about those drugs that the algorithm has correctly predicted and that were also experimentally validated and confirmed in biological labs? Can we see if there are certain common biological pathways or common genes or common network motifs or commonalities in their mechanisms of actions for those drugs, as that can lead to new insights? Now, to start asking these questions, we can take a variety of different approaches. We are very interested in ways that can explain machine learning predictions. And so in this particular case, techniques such as GNN explainer are algorithms that can be used with any graph neural network to explain individual predictions made by graph neural network. And these explainers can summarize where in the input data the model looks for evidence for its prediction, and they can identify an important part of the input graph, the input data set that drove the model to make a certain prediction. So let me just show an example of explanations. So in this example, a domain expert might ask a query, and they might be interested in knowing whether rosuvastatin would treat hyperlipidemia, and what could a disease treatment mechanism be for that form of statin inducing positive therapeutic effect in hyperlipidemia. So what an explainer would return, it would return a small subgraph, such as the one shown in the right part of the slide, that consists of a set of genes and a set of biological pathways that the algorithm deems most counterfactually relevant and for this particular prediction. And so that is an exciting opportunity that this new class of explainable models allow us to achieve so that predictions are more actionable. Going back to our COVID work, we used GNN explainers to try to understand, well, what is the possible mechanism of action for those novel drugs that the algorithm identified. So that study revealed that 76 out of 77 drugs that successfully reduced viral infection and were predicted by the algorithm were drugs that do not bind to proteins that are targeted directly by the virus. Instead, those drugs rely on network-based actions. Their primary targets are proteins that are somewhat far away from the viral targets in the interactome, and the way they exert their therapeutic effect is through a cascading series of non-direct interactions. So this is really an important point to make, because it indicates that having models that can take into account the structured network-based relationships was crucial to be able to identify this group of drugs, which we refer nowadays as network drugs, that would not be possible by alternative docking-based strategies. Okay, so to summarize this part, I illustrated a particular study using graph representation learning for rapid therapeutic innovation, and the two algorithmic techniques that allowed us to tackle that challenge was the ability to train models where labeled data is scarce, which is possible by few-shot learning, and the ability to understand predictions that were made by these models, which is possible through explainers and explainability methods. So going to the second part, which is a bit shorter, I will change gears a bit, and we will still talk about structure and knowledge, but we'll focus on a slightly different problem that is concerned with modeling effects of drugs when patients are already taking them. So it is quite common for patients to take multiple drugs at the same time, to be co-prescribed multiple drugs to treat complex or coexisting diseases. Indeed, statistics for the United States is such that 46% of people over 65 years take more than 5 prescription drugs, it is not uncommon to encounter patients that take up to 20 drugs to treat heart diseases, depression, cancer, some other complex diseases. And the problem with that is that the more drugs an individual patient is taking, the higher chance is that they will develop unwanted side effects. And those unwanted side effects affect 15% of the US population and incur an annual cost that exceed $177 billion in the US alone, because those side effects then need to be treated and they present a burden for the healthcare system and individual. When I said that patients taking multiple drugs at the same time are at much high risk of adverse event, that I meant the following. Those adverse events are due to unexpected interactions between the drugs that the person might be taking. So imagine this simulated example. We have an individual that is prescribed 2 drugs, a blue one and a red one. So when that person takes blue drug alone, everything is okay, there are no side effects. When a person takes red drug alone, everything is okay, and again, no unwanted side effects, adverse events are experienced. However, when a patient takes red and blue pill together, then with some probability, they experience side effects. This is unexpected. If the red and blue pill would not interact with each other somehow, then we would not expect to see any side effects. If a red and blue pill would act independently of each other, then given that none of them, when used alone, lead to side effects, we would not expect to see any additional side effects by them being taken together. However, we see those side effects in person. And so that is unexpected, and we would like to get rid of that. So that immediately creates a question: can we design a model that can predict whether a particular combination of drugs can lead to a particular type of side effect? And so to answer that question, the first step to take is to build a data set that would allow us to study this question. And so together with the US FDA, which is a regulatory body in the US taking care of food and drug approvals, we built a large polypharmacy knowledge graph, which is a graph that consists of drugs, their information about what targets those approved drugs have, and how those targets proteins interact with each other. And so the important part of information here are these triangle nodes, which are drugs, and 2 drugs are connected by an edge. That edge indicates that there is a specific type of adverse event, drug interaction that occurs when a patient takes both drugs, both represented in those triangles together, that we could not attribute to either of the 2 drugs alone. And so with that information about known drug interactions, can we use this polypharmacy graph to predict new interactions and early on identify possibly unsafe combinations of drugs? So the approach that can do that is called Decagon AI. And what Decagon AI can do, it takes as input this polypharmacy knowledge graph that we have seen on the previous slide, and it will map through, by specifying a graph neural network algorithm, it will map each and every drug in the input graph to its embedding vector in the embedding space. And it will do so such that once we have those embedding vectors of drugs, we can combine them to predict drug interactions that 2 drugs might have. So from the standpoint of a user of a Decagon AI model, a user might ask how likely it is that a form of statin and ciprofloxacin, these 2 drugs, when taken together, would lead to breakdown of muscle tissue. And what would happen in the backend? Decagon makes a prediction that is about that predicts probability of breakdown of muscle tissue type of link existing between simvastatin node and ciprofloxacin node in the knowledge graph. And it does that by combining those embedding vectors for the 2 drugs in the latent embedding space. Once we have those predictions, we can, of course, start readily examining them. And I'm showing here just one tidbit, one quick example of how those polypharmacy side effects associated with drug combinations can be evaluated. In this example, the Decagon AI model was trained on all data known and available up to 2012. And then we asked, well, how many predictions that the model made trained on data up to 2012 were confirmed later on, after 2012? So the table that I'm showing on this slide is showing top 10 predictions made by the model that come in the form of a pair of drugs and a side effect predicted by that drug. And for many of these cases, it is possible to find direct evidence of predicted interactions between drugs that were later found as unwanted side effects in real-world patients. So we did a variety of follow-up studies. Most recently, we were looking at over 10 million reports of adverse events for the entire US population across different age groups and different demographics information in order to identify those adverse event signatures of patients and identify what are patient populations that are at higher or lower risk for developing unwanted adverse events. So finally, I would like to just very briefly conclude with a resource and an initiative that really provides tools and libraries and benchmarks and data sets to a broader community that can then start and develop models, evaluate them, and deploy them in clinical biomedical settings of the kind of nature that I've highlighted in this talk. So this resource is called Therapeutics Data Commons. It is available at tdcommons.ai, and it presents a machine learning artificial intelligence foundation for problems related to therapeutic science. It serves as a meeting point between domain scientists on one end, biochemists, biologists, clinical researchers, who identify meaningful tasks, meaningful data sets. And on the other side, AI machine learning scientists, bioinformaticians, computational biologists who can design powerful computational methods. And working together in TDC, it's possible to facilitate algorithmic scientific advance in discovery of safe and effective therapies and identifying what methods work best, are most suitable for drug discovery applications and why that would be the case. Therapeutics Data Commons supports the entire life cycle of machine learning research that goes from formulating meaningful machine learning tasks to identifying, retrieving data sets, developing powerful models through connectors that go to PyTorch, TensorFlow, JAX, Hugging Face, other powerful deep learning frameworks, then to evaluating performance of machine learning models and comparing them against state-of-the-art models. And to do that, Therapeutics Data Commons consists and provides many AI-ready data sets that are as easy to retrieve as writing a couple of lines of code, methods that can evaluate performance of trained machine learning models in a systematic and transparent manner, in a manner that is indicative of real-world situations, and also provide leaderboards and benchmarks so that it's possible to define best-in-class model for a specific scientific question that can then be implemented in biomedical or clinical settings. So it's an open science initiative that I very much invite everyone here on the call to check out. And if you are interested in contributing to this initiative or using it, you are more than welcome, and you can reach out to our team through tdcommons.ai. So with that, I will wrap up for today. Final point I'd like to make that is that papers, codes, data sets that I've highlighted throughout this talk are all available from our lab website. I'm also very happy to take any questions in the remaining few minutes. And thank you again for inviting me to give a talk today here.
Thank you very much, Dr. Zitnik. Can you hear me? So yeah, it was very impressive and I think mind-opening presentation, and thank you very much again. We have one question from the chat box. I can read for you. Given the sensitive nature of drug development and clinical trials where human lives are involved, rigorous research is critical and it will be hard to compress the years into months. Nobody really wants to rush production for a product where people could die if the product is not well tested and validated. Could AI really shorten the years into months?
Yeah, so this question, that's a great question. And there are areas of drug development that should absolutely not be shortened, or kind of we should not cut borders for them. The project that I focused on has aimed to improve efficiency of preclinical drug discovery and development. So here in particular, those phases of early drug discovery and development that do not involve clinical trials yet, that are concerned with identifying the most promising chemical compounds, most promising drug candidates that would maximize the chance of developing then drugs that are safe and effective before they're being treated in patients, so that we can early on decide which of the compounds are such that research should be proceeded with them versus those that research should not be proceeded with them. That's where I see current existing AI methods to be most useful for. For late stage of drug development and involving clinical research, there are these important additional considerations. I did not cover them in this talk. I briefly alluded them as an important point that makes biomedical data unique in terms of these are decisions that involve human patients and human concerns. So any model would need to be not only accurate, which perhaps is the most important objective for the early stage discovery, but for late stage discovery, there are other concerns regarding trustworthiness and ethics and fairness and so on that I was not able to cover. So given that still the development of a new drug from scratch takes over 12 to 16 years on average, shortening any phase of that process, including non-clinical phases, can substantially improve efficiency and faster and decrease the time needed to put a new drug on the market.